Pregnancy in mammals is a highly regulated process involving a multitude of physiological changes, including distinct effects on the brain throughout gestation. These changes range from macroscopic differences in gray and white matter volume, ventricle size, and fluid regulation to altered neuroplasticity and specific molecular changes in neuropeptide expression and signaling. Throughout pregnancy, these processes both support gestation and also prepare the female for motherhood by driving cognitive and emotional adaptations that promote maternal behavior. We explore these neurological changes with Illumina's spatial technology platform. Our platform provides high-sensitivity gene expression, whole transcriptome coverage, and high-resolution spatial information. So in this experiment, we compared a virgin female mouse to that of a pregnant, late-term pregnant mouse. And so we dissected the brains and flash froze them, and then placed the coronal sections on our Illumina spatial substrate. So one aspect of our product is a very large spatial substrate, so 15 millimeters by 50 millimeters. And you can see in this case, we were able to place 4 sections on each of these substrates pretty comfortably. We also looked at 2 levels within the brain. So we studied the preoptic level and also the second level targeting the hippocampus and hypothalamus. And we looked at 2 sections from each mouse at each of those coronal sections. So here's a look at our workflow. We start with a pretty standard histological workflow. So we do cryosectioning and then H&E staining and imaging. After that, we go through our spatial library prep. So this includes tissue permeabilization, RNA capture, RNA capture, and then it comes off of the flow cell. And then so from there, it goes through standard library prep and sequencing. In this case, we used a NovaSeq. You could also use a NextSeq. And then the data is analyzed through Illumina Connected Multiomics, which is our software, which includes our optimized spatial DRAGON pipeline to look at this vast amount of data, and then also a visualization tool, and our software also supports tertiary analyses. So here's a look at some of the raw data. You can see the H&E images at the top and the spatial heat maps at the bottom. In this case, each of the 2 slides that we have, we looked at about 700,000 cells on each slide. So the 2 slides were about 1.5 million cells in total. And we're able to see about 1,000 unique transcripts per cell. And you can see the sequencing saturation plots and also the violin plots on the right of the slide. Interestingly, we also were able to capture over 30,000 unique transcripts in this data set out of the 60,000 total genes in the mouse DRAGON reference. You can also see really good reproducibility between the samples and also across the substrates. So here's a look at the Illumina Connected Multiomics visualization tool. So this tool is able to look at the H&E image and overlay the spatial transcriptomic data on top of that. It also supports tertiary analyses like light and clustering, and we're able to do cellular segmentation. To the bottom, I'm showing a couple of single genes. That's another thing the software can do, is look at single genes. So in this case, 2 of the glutamate receptors, vGlut1 and vGlut2, and so these are markers for separate populations of excitatory neurons, and we see the expression of these genes specifically located to the expected areas. We're also showing the myelin basic protein, which is correctly localized to areas rich in oligodendrocytes, as expected. On the right is kind of a zoom in of some of those Leiden clusters, and so showing 2 there, the lateral ventricle and the medial habenula, and you can see they're enriched for a specific cell type, so the dark blue clusters making up the lateral ventricle and the dark purple making up the medial habenula. And then to the far right are some marker genes that are associated with each of those clusters, and you can see they're all co-localized as you would expect. So the data is also compatible with third-party tools. So you can export the data out and use any third-party analyses tools that you might be comfortable with. In this case, we combine the spatial transcriptomic data set with published single-cell reference data in order to do the cell annotation. You can see on the right all the different annotated cell types and the nice images of the brain as they come together. So you can see the cortical layers there in the pink and teal, the hippocampal layers there, turquoise. The hypothalamic inhibitory neurons are clearly there in orange, and then the white matter is in dark blue. So we're able to generate these really nice high-resolution spatial heat maps. Okay, so then from there, we wanted to try to highlight the discovery power of a tool like this. And so we're just going to take a look at the data from a few different angles. So we started here with a spatially driven approach. And so the first thing we did is a differential gene expression analysis. So that's on the left. The x-axis is all of the annotated cell types that we just looked at. And then we looked at differential gene expression of the pregnant brain relative to the virgin brain. And then you could also visualize that differential gene expression as a heat map, and so that's in the middle. And a couple areas jump out to you. So, for example, level 2 there, you can see the red region, which is the Habenula nucleus, and so that's lighting up as a really hot spot for upregulated genes in the pregnant mouse. But there's also other areas there, like the thalamus and some of the cortical layers. And then on the right, we're trying to highlight the value of this whole transcriptomic data set. So as I mentioned, we're seeing over 30,000 unique genes in this data set, and of those, more than 2,000 are differentially expressed. And you can just contrast that to a small panel type of approach. So for example, a 500 gene panel where you would have only seen 66 of those differentially expressed genes out of the more than 2,000 that we saw. So taking a closer look at one of those areas, like the Habenula, we did a zoom in of the cellular neighborhood there on the left, so looking at an area a little bit larger than a square millimeter. And the habenular cells are clustered there in the dark green. And you don't see any striking morphological differences there between the virgin mouse and the pregnant mouse, with the exception of the more prominent choroid plexus in the light green, which we'll come back to later. But continuing on looking at their habenula, we then exported this data to a third-party tool called CellChat2, which is able to look at communication pathways by looking at ligand receptor signaling pathways between neighboring cells. And so that's visualized in the plot on the center with all of the cell types around the circumference. And the lines there are the communication pathways between the cells, where the red lines are upregulated communication pathways and the blue are downregulated communication pathways in the pregnant brain relative to the virgin brain. And if you look at the green habenula there at 1 o'clock or so, you can see all of the red lines coming out of that, just highlighting all of the increased communication pathways that's happening with those cell types. And then you could take that even a step further and look at the different molecules that are involved in those communication pathways. And so that's on the right. And you can see the habenular column there. There's a number of molecules that are overly expressed in the pregnant brain, again, relative to the virgin brain. So just an example of all these different areas that you could dig into and find new discoveries with a system like this. Next, we took more of a histological approach. So coming back to that more pronounced choroid plexus, and you can see that in the H&E image, there's a much higher density of choroid plexus cells in the pregnant brain relative to the virgin brain. And you can double check that with the spatial image there that's also shown. And again, you can see the cells are clustering really nicely as the light green choroid plexus with kind of a cell wall there in blue, the endothelial cells making up the cell wall. And again, you see the higher density of cells in the choroid plexus. So then we took a closer look at that, did a differential gene expression analysis on the choroid plexus there, and decided to take a closer look at this one prolactin receptor. So prolactin is a hormone that stimulates mammary tissue growth and lactation, but it's also known to have local effects in the brain. So to further investigate whether downstream genes involved in the prolactin signaling are also enriched, we decided to perform a gene set enrichment analysis. So that's shown on the right. And so we see enrichment in the prolactin communication pathway, but also in several other downstream pathways. And then interestingly, on the bottom panel on the right, each of those vertical lines are different genes that are involved in that signaling pathway. And you can see, again, the very high resolution of genes that we're able to get, which is a function of the whole transcriptome data set that we were able to generate. So from there, we took a cell type of interest angle to look at it. So it's well known that there's significant gene expression changes in white matter in the pregnant brain, and white matter is primarily made up of oligodendrocytes. So we took a closer look at the oligodendrocyte cell cluster, again, did a differential gene analysis, and we just chose a couple of genes here. So SGK1 is a kinase known to be involved in cell stress and survival, and IL-33 is an interleukin with demonstrated roles in brain disease and injury. And we didn't find any literature on either of these two genes, so I think these are relatively new. And you see the SGK1 is overly expressed in the pregnant brain relative to the virgin brain, and IL-33 is underexpressed in the pregnant brain. And you can visualize those as well in the plots on the right. And then finally, we took a gene of interest approach. So here we took a look at oxytocin and vasopressin, which we know are neuropeptides with important roles in regulating social behavior. And so we see that we found that these are highly localized to the hypothalamus region, the paraventricular nucleus in the hypothalamus. And so put a little zoom in of those there. And you can see the pregnant brain, they're much more highly expressed than in the virgin brain. And we were able to verify that with the differential gene expression plot. And then also it's been previously shown that these neuropeptides are not commonly co-expressed in the same cell. And so the fact that these genes don't co-localize in our assay highlights the high resolution and spatial specificity of our assay. So you can see the little red and blue dots there. They're not overlaid on top of each other, but they're slightly shifted relative to one another. Okay, so hopefully I've shown you some of the features of our system. So the high sensitivity gene expression data that we're able to generate, the whole transcriptome data, the high spatial resolution. So some of these features we think are going to be really powerful for promoting discovery and especially spatial discovery. And we're really excited to get this tool out there into your hands and see what people can do with it.